So I would give an overview of our research here, what we are doing in our group. That's why it's titled Structural and Functional Network Characterization for different neurological disorder patients. So first I give you some introductory slides and then go on with some real application of data, because what we do is that we try to analyze these data and then we go into the application of helping the clinicians basically to identify some markers for their clinical therapy. So speaking about clinical therapy, what we do in minds is that we call this therapy a deep brain stimulation. So here's a picture shown below that you have an electrode which is sitting inside the brain, and the person, for example, in this case, a Parkinson patient who has problems, for example, like tremor of the hand, they will be implanted with an electrode in the brain, and then it's being connected to this stimulator, which is then sitting under the shoulder bone, and they are then stimulated 24 hours, so that the good thing about this type of therapy is that they don't have then this tremor anymore. So this is done very often, and this is some DBS electrode. How does it look like? So you just give you an idea how big it is. It's about 7.5 millimeters are the contacts in this electrode. It can be in any way stimulated, and once you stimulate the brain, then you can also model how the stimulation field looks like. And there are, of course, additional developments. You have directional leads, meaning that you can project the current to a certain part of the brain, and non-directional, then you have a circular kind of current. And what we do is that when we have this active contact location, as we call here in the brain, then we look at different connectivity fingerprints. So we differentiate this connectivity into two: structural, meaning anatomical connectivity, which is present in the brain, and functional connectivity means that you do a certain task and then some regions light up and some regions don't show any activity. And then we can, of course, in the case of structural connectivity, we can isolate these running fibers from where we are stimulating to other regions in the brain. And using these fingerprints, we can either associate to the, let's say, to the voltage, how much we are stimulating, or any type of clinical score, how bad are they in this disease. We can either do an association or we do a prediction, to that at the end we give an individualized therapy to each of these patients. So I give you a few more introductory slides to give you an overlook of what kind of data we are having. So this is a slide where we show, for example, if some of you don't know about the regions in the brain, so this is the region in the brain which we are stimulating, which is very important for motor tasks, the hand or for the leg, and how it is connected to other regions in the brain on the top of the brain, and this is very deep in the brain. So we have several different modalities. Probably some of you have heard about it. So tractography means the diffusion tensor imaging, how does it, the tracks are running anatomically, or functional magnetic resonance imaging, we look at functional data, how is the bold, meaning how does the blood oxygen go up and low, or we have positron emission tomography, and we do a lot of EEG, meaning electroencephalography, and you can look at these connections. Most of these modalities are able to find those connections, but there are very subtle differences in these connectivity. So the good thing about this type of operation is that we are putting an electrode inside the brain, which is commonly not possible. Usually it's only done in mice or other animals, but in this case we have the capability to put in the human electrode, and then we are able to also record directly here. So basically neurons, you can record them directly. It can be single or multi-unit. Or you can also record, let's say, local field potentials, which is a summation of all the neuronal activity. Or we have the capability to have a electrocartographic grid. What it means is we put a strip of electrodes on the top of the brain, and then we can also look at local field potentials. Or we can directly on the top of the scalp, we can put an EEG electrode and record the surface activity. So these are some of the methods what we use. We call them electrophysiological proxies. So just go into one or two of these measures. Let's say coherence, which simply means correlation in the time domain. Here in the frequency domain, we just want to know which frequency is present, let's say, in the EEG or in the hand movement. We can also measure kinematic senses, what is the movement or muscle activity, and then we can look the coupling between this hand and the EEG electrodes. Or we can look at, let's say, phase amplitude coupling. This is also a type of coupling which relates the phase of one signal to the amplitude of another signal. And of course, probably other techniques are known to you, like power spectrum or looking at in the time frequency domain. So we first start with the structural domain. So this is an MRI data what we get from the patients. So this is done before the operation. And then what we do is we try to disentangle each of these regions and then try to estimate something called cortical thickness, which simply means that how thick is the cortex in this person. And using this parameter, we then develop correlation matrices and then use this for either group comparisons to see whether there is a group of people who have a very good outcome after this DBS, how is the thickness in that group compared to a group where there is very not that good outcome. We look at network topology and use this also for the comparison, and finally feed this all into support vector machines and try to give us a good prediction so that we can use this to say to the clinician whether this particular patient will be able to profit from this type of therapy. I give a quick introductory slide about graph theoretical analysis, because in this study we use this type of analysis. And here the brain is divided into nodes, and the connections, we call them edges. And we try to build the whole brain in a type of graph. And then using this graph based on the assumption what we have, either it's a single region or two regions or many regions, we then of course go to a multivariate analysis and then look at the network, how good is the network compared to a patient to a healthy control, for example. So we looked at two different group of patients. We call the superior outcome means they profited well with this deep brain stimulation. Moderate outcome means they are not very well profited. So we want to see the difference, what happens in the brain. And these blue regions are the regions which are then difference between the two groups. And some of the regions, if you don't know what about brain, then meaning here is the frontal part of the brain, they are responsible for a task. And for example, here the occipital part, meaning it's responsible for the eyes. So these are regions which are highlighting that there are differences in these two groups. And we use this to develop some network parameters. We call them clustering coefficient in this case. It's simple, it's very similar to the clustering algorithm, what most of you know. So this is a region in the brain, and if it had connections to all the neighbors, then you have a low. And if you have additional connections here, the clustering coefficient is high. And the same way as degree, low means less, high means higher degree. And we again look into these two groups and look at this degree and clustering and find out regions which are significantly different. Again, colored here, they have low degree, and the CN color here, for example, have high centrality or high degree. The same way is plotted for clustering. So once we identify these regions, then what we do is we fit that to support vector machines, and in the violet is shown for the network selected regions, and for the yellow or orange is shown for the volume at deeper in the brain. and we see how good we can classify these two groups. We achieve about 75% accuracy. And always in the clinical point of view, if you want to make the clinician believe, then you need to train your algorithm in one of the groups. That's why we call this discovery, and then test this algorithm in an independent group to show that this algorithm works. And there we, for example, achieve about 80%. And here are the receiver operating curves and the area under the curve. So each region, even if one single region we are able to take here, we can get an accuracy about 82%. So this is looking into the cortical thickness, the thickness of the brain. We also look in these patients how this tractography is what we call, we image the brain with a special sequence, and then we can have these nice pictures how good the tracks are going from a specific region to other regions in the brain. And then, as explained before, we again develop a connectivity matrix and then look at several regions in the brain, how good they are connected. So here again, we use this frontal part of the brain, these connections which are shown here, and these circles are regions. And then we use this for prediction. And again, you can see, for example, in the frontal region of the brain, you get a very good prediction of 82%, meaning we can use the patients even before they're going into the operation theater, how good they are able to uh, profit from this type of treatment. So this is very helpful for the clinicians. Now I move my focus from the Parkinson disease to multiple sclerosis. So these are patients who have lesions, we call them, in the brain, which is then not functioning really well. So these white spots here are called lesions, which means they are not doing the function what they're supposed to do. So what we want to find out is that this topography of these lesions, as shown here, do they go well with the clinical parameters, what we can measure, whether they are progressed in their disease or do they are in the beginning. Can we predict this disease progression using this lesion topography? Here we used independent component analysis as shown here. We use structural images and then look at the shared contribution between the lesion topography and we call the atrophy, which means again the cortical thickness, how does it change from one time point to another time point. And then we can look at different ICA or independent component analysis patterns. So we found 3 different patterns. This is a deeper region in the brain, and these are regions which are higher in the brain. And when we find these patterns, whether these patterns are useful to classify the disease progression. And here on the left side, we show again the receiver operating curves. So this is a pattern which is identified in the cerebellum, which is a region which is very important for any kind of action what we are doing, for example, the arm movement or the leg movement. And here we can see that the cerebellum is then giving us, as shown here in green, gives the best accuracy in predicting the disease progression. And here is a connectivity map. How does this pattern is related to different of these regions in the brain, shown in different colors. And what we did is we went one step further, taking this cerebral pattern and then fed that into convolutional neural network, and then try to find out whether we get good accuracy with this cerebral pattern. So again, here we show for the 3 patterns which I showed before, and here is the axis shows the prediction accuracy, and here are the number of subjects. So we used 50% of the subjects to do the model, and then use the other 50% to do the classification. And here, for example, again, the cerebral pattern, we get an accuracy of about 95%. And this is then predicting how progressed they are in this disease stage. And again, as I said before, we need to always show that in an independent cohort, so that we then used another cohort of patient and then did a similar analysis. There we found only 2 patterns, but still above 90%, we were able to classify the accuracy, classify the patients who were progressed and who were not progressed using this cerebral pattern. So these are studies which I showed on the structural and the domain. And now I move on to the EEG analysis. What we do is we call this an inverse solution, because what we record in the brain is EEG or MEG data. So this is how the EEG electrodes looks like on the brain with these blue circles. And then the MEG coils are shown here. And what we get is we get time series of a certain amount of time, like say here 2 seconds. And then what we want to know is if we do a certain task, let's say if we ask them to move the finger, we want to know which of the brain regions are active. So for that, we just have this scalp data. We want to know from where this signal comes from. So that's why we call this inverse. So we have this data and we want to find this. And what we do is we call this beamformer analysis. And this beamformer analysis is similar in other domains. Probably if you know, like for example, radio frequency domain, there also they use this beamforming, and then we try to locate this source. We always use the EMG, that's the muscle signal, as our reference to locate the source. And what we do is we trick the algorithm to say, first find the most activated source or the strongest, and then we tell the algorithm in an iterative way to do as a black box and then go further with the other sources. So in this way, we will be able to get the other sources, even deeper regions in the brain. The nice thing about this type of analysis is you can also get a signal out of this source as shown here, and then you can do also connectivity between these signals. Here I show the connectivity analysis, what we do generally in our data. We call this effective, meaning causal. So we want to know the causal information. Here I show a simple model simulation. So we have 3 time series from the brain. And what we show here is this time series X2 is giving information to X1, and the same happens with X3. And they have a time dynamics. So this connection, which is shown here, has a waxing sine wave, for example, or the X3 is a triangular wave. And we want to know this dynamics and also know the causal connectivity. In some algorithms, what we get is you get some very high values. For example, let's say Granger causality, then you get high values here and other values are lower, which means it just shows what amount of connectivity is being between these 2 signals. But we wanted to also look at the frequency at which is happening. Then we also get the frequency information. And what we finally do is we also look at the time domain. So at the end, we also get this waxing sine wave and this triangular wave, and all the other connections are 0. So there is no connectivity in these time series, but in these 2 time series, we get a very good connectivity estimate. So we use this type of analysis to again differentiate between patients and healthy controls. So as I showed the pipeline earlier, EEG, EMG, the coherence, and then we do the inverse solution, and then estimate that in a certain atlas, which gives you good connectivity maps. So here is Parkinson's disease, who have a problem with the hand, let's say tremor, and there is another tremor group, and here is the healthy controls, which means they imitate the tremor in these subjects. And then we find subtle differences in certain regions of the brain. And then we can also find, using this analysis of connectivity, to show which are the differences. So this is healthy control, and these are the patients. So you can see there are differences in the connectivity, because you can use this information then for stimulation of these patients. In another cohort of patients, as in another study recently, we were able to show that these are, for example, the electrodes which are sitting deep in the brain. And these are 2 groups of subjects where we stimulated in different frequencies. And here is again the electrode. And as I said before, how much of the tissue is activated can be modeled. And then we look at the correlation between this and regions in the brain. And then finally, we looked at cross-frequency coupling, meaning that how one frequency is dependent on another frequency. And then we were able to show that in different regions in the brain, so this is a certain oscillation. If some of you don't know, beta is around the frequency of 13 to 30 hertz, and gamma is from 30 to 100 hertz. And here is the stimulation, what is used generally in the clinics. And here is off, so we switch off the stimulation. That's really nice in this patient. You can just switch off and the stimulation. And we also use low, means we stimulated 20 hertz lower, and high means we stimulated 20 hertz higher. So you see this clear dynamics here in this group of patients. So there's a reduce in this beta and an increase in this gamma. So this we can find in most of these regions in the brain. And this region was used as a control region to see whether this doesn't happen. So it doesn't have any dynamics. So this is true what we are seeing in these other regions. And of course, what we are measuring is EEG on the top of the head. And I'm showing you regions which are very deep in the brain. So to validate this, we also were able to record directly from the deep of the brain and the EEG simultaneously. And here we show a time frequency graph. This is time, this is frequency. And you can see the dynamics of this coherence, so coupling between the brain and the muscle, have a similar dynamics for the recorded signal and the reconstructed signal. So this holds true and shows you that the analysis what we are doing is validated by this type of analysis. And using the next 3 slides, I just show in our unit what we are doing. So this is what we are trying to do. We use all these different modalities. We bring that to a network perspective. And we also use, for example, mouse data. And then what we want to do is build predictive models, and then think about critical time windows where a therapy would be useful. And now, as we've seen a lot today about other things like gene expression, proteins, so this is also one field we are touching in new, trying to find out also these kind of modalities to develop them as matrices, and then develop as network parameters, and then look into deep learning algorithm to see whether we are able to use this to also for clinical use. And finally, I think what we want to achieve is shown here. So we use all these parameters, we identify different network changes, and we use model approaches and deep learning approaches. And then finally, if we can give the clinician a therapeutic option, let's say either invasive or non-invasive, or even at some point we can say we could change lifestyle modification, let's say more sports or stop smoking, for example. So this kind of things can help the patients in a way that they can then try to go ahead in the disease what they have and not to progress. Yeah. So this is my final slide. So of course, all this work is not possible with a great group of people. So we have 5 PhD students, and they are 2 of them already left. They finished with us, and they are in US now. And we have clinicians, and we have technicians. So it's a group of people very highly dynamic, and that it's possible to do such kind of analysis. And here are the collaborators, and here are my sponsors. Thank you for your time, and I thanks also for the organizers to giving me this opportunity. Thank you.